Here we are again. Hi, hello, and welcome again. Yep, I can hear myself. Hello, and welcome again to another Saturday live stream. I'm Oliver. Happy that you could join me again. Just give me a second or so to get all of the technical stuff sorted out first. Sound, I hope, does seem to be working. Okay, so I think we're ready to go and to roll again. Yes, again, welcome around the world. Unlike the previous years, I'm not going to call out every individual greeting on the chat. I just want to say a general hello all around the world. Wants to keep the live stream also a little bit shorter than in the previous years. I would like to do a live stream again, 1 hour, and keep it a little bit more compact. And I have another short editorial announcements to make, and that is I sent out, or I'm sending out today, the March newsletter. So with a couple of ideas on there as well. Usually the sending of the newsletter takes several hours because the 1,300 subscribers that I have currently. So if you're interested, then you can of course also join and on the website, on the microbehunter.com website. Basically, that's one thing. And another thing is, yes, unfortunately, next week, I'm also not able to do a live stream again. But then afterwards, again, it's basically I have some other arrangements again. What I will do is the following. I will please feel free to ask questions and to comment in the chat. In order to keep the whole thing a little bit more streamlined, I will kind of not answer the chat questions right away, but I would like to concentrate them again and try to talk, to answer them later on. Okay, but please feel free to comment in the chat. Today I would like to do three things. I would like to look at three things. I'm a little bit flexible as always, of course, but one thing that is kind of nice, nothing entirely new, but over here, this is Elodea or Egeria densa. It's a water plant. A colleague of mine in school, I'm a teacher, as you know, he's got an aquarium and he kind of cleaned out his aquarium and he gave me this, these water plants. It's way too dense, as you can see here, and you have to put it into a larger jar, but this is a very nice plant to observe something called cytoplasmic streaming. So we're going to have a little bit of a look at this. Actually, one of my more favorite specimens. I'll talk a little bit also about biology here, okay? So I'm going to combine it with a slightly light biology lesson. So that is number one. Number two, look, you won't be able to recognize this anymore, but I collected a water sample from a pond, I think it was 2 weeks ago, 3 weeks ago, and I treated it with hydrogen peroxide. I started this last week. So if you joined in last week's live stream, then you basically remember when I put some hydrogen peroxide over it. This is how it looks now, right? It looks completely clear, and it looks something like a sand, almost like sand over here. There are a few diatoms in here. This is not the nicest sample. I've had also much nicer samples before. But I just want to show you a little bit the proof of concept, how to extract diatom cell walls. And last but not least, what I have over here, this here is vinegar. And this is a little bit old and there's some stuff floating in here. So these must be some bacteria. And I would like to put those also under the microscope. Some people call it the mother of vinegar. It's an interesting term. But these are basically bacteria that are able to grow in acid. Okay, so this is basically the thing that I would like to look at today with you. Yes, again, feel free to comment. I always have a look with one eye. I'm always looking at the comments as well, but I want to respond to them maybe in a slightly more organized way than in the previous live streams. So let's get started with this one over here, and we're gonna do a real. We're gonna go, of course, also using. Oil immersion here. We're going to go to the maximum magnification. I kind of really like this specimen a lot. So how do you proceed here? Well, it's the easiest specimen to prepare, because all you do is you pull it out here. You see the top ones are a little bit dried up already here because the water evaporated. So you want to get one further down, and all you do is the following: you just rip off a leaf here. That's it. Oh, where is it? lost it. Rip one off here. Yeah, and it's kind of yeah, large. So what I would like to do is I'm going to actually cut it. Here is my scissor, because otherwise if it's too large, then we have the problem with the cover glass. So if you go do it like this, and generally this works. Yeah. Again, because I just have seen that somebody commented, super excited for the diatom shell extraction. Don't be too disappointed, okay? Please. It's not the nicest specimen. It does kind of, but it kind of shows the principle, okay? That's why I've done that. Hydrogen peroxide extraction is actually a good way generally, but I think the specimen in this case was not the best one. Yeah. So obviously I need a little bit of water. So what I'm going to do now is add a little bit of water. Here is my pipette over here. So I'm just going to take directly some water from here and add a little bit of water here. And cover glass goes on top, and we already know that. Lup. And that's pretty much it. I think one of the. Let's turn off the light here as well, otherwise it clears. I think it's really one of the easiest. Ooh, the cover glass is broken here. Doesn't matter. Yeah, it's really one of the nicest, easiest specimens to prepare. So let's have a look. Okay, as always, we start at the lowest magnification. I have now also. Yeah, look, I bought myself something new, which makes switching easier. In case you're kind of wondering. This is what I've got now, and by pressing the button here, I can switch easily, the display. So that's I can switch back and forth. That's makes live streaming much easier for me. Okay, now we are here at the microscope view, and let's put it in here. And this is how it looks like. Okay, so. This is the reason why it's such a nice specimen is because the leaf is extremely thin and transparent. Okay, so of course a bubble over here. This is where I cut it, yeah. But of course we want to really do we want to really zoom in quite a lot. Okay, so. I'm also seeing over here that somebody's commenting about the time zone. Okay, just for your info, I know that I think in the United States there has already been a change in summertime, daylight savings times. Here in Europe, where I am right now, look, there's a little protozone over here. I think it has not happened yet. So basically, it might be adjusted again in a few weeks. I think end of March we have to change the summer savings time here. Yeah. So, and I know it's not the same in the United States and in Europe. That's why if you're in the US, then this is going to be at a different time than you probably are used to. Yeah. So by the way, also look here. What I have is the following. I want to explain this as well to do a little bit of technology as well. Look, this. Let's go back here. This is now the 4x objective. Okay, so the low power, and right now, and look what happens to the condenser. I can swing it in and out. Okay, and when I swing it in like this, then you see that it does not fill the full frame. Yeah, and for this reason, there is the option to swing it out. Yeah, so that you actually get the full frame here, right? Yeah, and then for all of the higher magnifications here, you see it's kind of dark, but then when you swing it in, it becomes significantly brighter. Yeah. So basically, okay. So this is the leaf at higher magnification. You can already see the individual cells, of course. I think I don't have to point that out to most of you. But what we really are interested in is basically, the chloroplasts. We have to find basically a space where we can see them actually move much better. Yeah. Look, if you're focusing now through the whole leaf, okay, it's blur, blur, blur. You see that the leaves, the cells go in focus, and I continue focusing, and then you see that another layer of cells slowly comes into focus as well, or is it not? Yeah, another layer here. Yeah, so this leaf seems to be probably around 2 cell layers thick. Yeah, so that's quite good. We move up. Okay, I'm bright field right now, so that's yeah. Some people ask me about the colors and yeah, because I've got this fancy equipment. I just want to tell you that essentially right now we're just looking at bright field, and if yeah, you should be able to see that with pretty much any microscope. Yeah. Ah, yeah, in Canada too. So in Canada also there is this change in yeah, different time. So we are now yeah, pretty close up with 60 times. Yeah, and we have to now look for a place where the chloroplasts are nicely visible. Yeah, and usually they're so dense, but over here, do you see here right on the side? Yeah, that's actually quite an interesting one. Yeah. And now I'm going to do the following. I'm going to yeah, now go to the maximum magnification. What is this thing here? Ah, okay. It looks like a diatom all the way on the right. Yeah. So let's now do the following. I'm going to now go to maximum magnification using the 100 times oil immersion objective, and now still without immersion oil. Without immersion oil, you see everything's much darker. Obviously, it's a little bit out of focus as expected. This is how it looks like. It's kind of blurry, right? Yeah. So what I'm going to do now is I'm going to add some immersion oil. I'm going to engage the focus lock. You might already know this from one of my previous live streams. Here I've got a bottle of immersion oil here with warning labels. Yeah, this is a synthetic immersion oil that I have here. It's totally clear. It's not yellowish. In many microscopes that you buy these days, they come with a bottle of immersion oil that's yellowish. That's a natural one. But they have the disadvantage, the natural ones, that they actually might harden. So yeah, I've engaged the focus lock lever, right? And basically I'm now using my coarse focus to turn it go all the way down here, yeah, to give it a little bit more space. Watch what I'm doing up there. I add a drop of immersion oil directly on the, yeah, directly on the cover glass, of course. And I close the bottle. Oh, it's pretty greasy. And I use the coarse focus knob again to raise the stage, and until the oil is dipped into the objective. Here we go. Okay. Then we can use the fine focus. And I think you see that this is way crisper and much better, right? Yeah. So this is yeah, actually quite nice. And yeah, let's move this a little bit more like this. Yeah. You can see that the green chloroplasts, yeah, are moving inside the cell over here, right? Yeah. So let me show this to you. Of course, it's not difficult to see. Here is the arrow. You see that these basically, yeah, these green things here are the chloroplasts. And what you might notice as well is that many of them are actually moving along on the side. Yeah. So this is basically a very nice demonstration of what we call so-called cytoplasmic streaming. And yeah, cytoplasmic streaming is can be observed yeah, of course, in many different cells, but it's very, very pronounced and easy to see in this water plant here. Probably also because it's so thin, and therefore the leaf is so thin, so that it's easier to see. Because if you put a thicker leaf under the microscope, everything's going to be completely black and dark because it wouldn't allow any light to go through. Okay, so we can also have a look here. Yeah, usually it's much easier to see in those cells which are a little bit not quite as tightly packed. So there's something else that you might notice, and this is also has occasionally been a question. If you see over here, this is kind of this jittery movement that's of whatever is in here, maybe some cell organelles and so on. We're already at such a high magnification that we're able to see something that's called Brownian motion. So that's basically a random motion of particles. Yeah. So this is something that is yeah, also visible here. I always look also at, in case you're wondering, I'm looking here, would be nice to also see the stem. We can do that. I need to be able to cut it somehow. I've a good point. I've never really looked at the stem of Elodea yet. we might actually do this as well. Okay. Actually, why not? Yeah. So let's have a look. So now a little bit to the biology part, right? Because this is chloroplasts are quite interesting. Of course, they're green because of the green pigment chlorophyll. Because what do? They do photosynthesis, right? So they catch sunlight, and they make sugar for the plant, and this sugar then is converted to cellulose and or to starch. Starch for storage, and the cellulose to make cell wall. And those chloroplasts are interesting because, they are moving, but they're not alive. And this is where it's getting really a little bit weird, okay? Because those chloroplasts, they're moving inside this cell, but they're not moving on their own. They're being pulled along by the cytoplasm, which is moving. And therefore, they're just basically passive, right? And are kind of just floating along with the other cell organelles that are inside the plant. So the question is now a little bit, and this is where it becomes interesting. It's a little bit also philosophical. I like to go a little bit into the philosophy of biology sometimes as well. It is because those cell organelles, per definition, are not alive. The chloroplasts, they are not alive, and not because they don't move on their own, but because they are part of a plant cell. So the cell theory states that everything that's kind of moving in there, right, inside here, all of this other stuff, those things on their own are not considered to be alive. It's only the whole cell that you see here, which basically qualifies of being called alive, right? So that's basically the cell theory, which one part states the basic unit of life is the cell, and everything that's smaller than a cell, even moving or not, right, is not considered to be alive. Now, where does it get philosophical here? This is kind of where it's getting really weird. It is because where do these chloroplasts come from? Because many years ago, a long time ago, when eukaryotic cells like you have here, when they first formed, those chloroplasts were independent bacteria which lived outside of the cell. So, they were once, so to say, alive because they were independent cells. As a matter of fact, we now know that they are actually related, very much related to the so-called cyanobacteria. Then those bacteria essentially went into a host cell and established themselves in the host cell, in the process called endosymbiosis. Endo means within, and symbiosis means that those chloroplasts, they benefited from the protection of the host cell, and the host cell benefited because now it was able to do photosynthesis and got food. So kind of a mutual relationship. And I remember when I first heard that, a long time ago, I thought, what? This is kind of weird. What? A cell living inside a cell? How can that be? Actually, it's not that unusual because this endosymbiosis is something that you can also find in protozoans and in other, it's nothing unusual really. And I also know of there are those some plants where even if you, and this is kind of interesting, even if you remove, if you kind of extract those chloroplasts, they're even able to live on their own. There are some plants out there where this dependency is not even that high. So even though it's kind of weird when I heard about it the first time, that those chloroplasts once were independent, it's not really that rare or unusual. And of course, scientists did some other studies as well, and they discovered that, of course, the chloroplasts have their own DNA even. So of course they did DNA studies and so on. So you see, it's interesting a little bit of what we're able to find out these days. Chloroplasts easy to see, and what they do is inside the cell they also divide like cells, right? So they also copy the DNA and they divide. There are a couple of questions here which I consider quite interesting about the white blood cell and so on. So this is the thing a little bit, is again, are the chloroplasts now alive? They have their own DNA, right? However, if you say the cell is the basic unit of life, and chloroplasts actually are a part of a cell, per definition they're not alive. But actually they kind of are, right? And even having their own DNA does not make it, that's not enough really as a criterion, because if you take them out, they're not able to live on their own. But in some cases they are. There are some plants that actually where it is possible. So why do I consider this so interesting is because it kind of shows a little bit that our definitions that we kind of invent as human beings, and the definitions and the categories and all these things, yeah, nature doesn't care about those, right? Those categories and terms and definitions are basically only in our head and nature just doesn't care, right? Nature does its own thing, so to say. It doesn't care whether we call it alive or not alive or whatever, right? No, it just does its own thing. So what we have over here, of course, also quite nice. This transparent round structure here. That's of course the nucleus of the cell. You can actually, yeah, here you can see it quite nicely because it's not so fully packed with chloroplasts. Yeah. So it's look at this. Wow, there's some movement going on here as well. Look, that's interesting. Yeah, and I made some time-lapse videos as well. And yeah, in case, I know that different viewers, some of them watch my shorts, some only the live streams. In case you're interested, just a few days ago, if you go into the shorts section, I published again one of those, moving chloroplasts in time-lapse, where really sped up, I was really speeding up the speed. Yeah, look at this. It's... when I remember a couple of years ago, I showed some cells to my students, right? We've got a whole bunch of microscopes and just gave them a microscopy assignment. They had to look at some ready prepared onion cells, right? I've looked at those hundreds of times already. But I remember that one of the students, when she first saw those cells, she was totally, I mean, it was not even it was a prepared specimen. It wasn't even alive, no movement or anything. But when I showed it to her, she was totally surprised and actually, yeah, stunned. And I asked her, "Are you okay?" Because she was overwhelmed. And I says, "Are you having problems with your microscope?" You know. And then she says, "No, everything's fine. And then because you're making such a frustrated face," I said, "You look so unhappy, right? Is everything okay with you?" And then she said, no, I'm totally fine. It's just like this. Yeah, on just such a small microscope slide, there is so much more there. I did not expect this. Yeah, that life can be so complex, right? That's actually this. That is really very, very true. Yeah. So. Yeah, so this is actually, you know what? Why? How am I supposed to do this? How am I supposed? That's an interesting one. I have to cut the stem now. Let's try this. Where is my? I need a very sharp knife. So let's try that. I'm looking for my razor blade because there's been the request to make a cross section of the stem, and honestly. Good point. I've never done this before. Really, I mean, of course I made stem cross sections, but of Elodea, I've never looked at a stem before. So this is actually a good, how do you say, thing to do. Let me quickly have a look at the. Here it is. Look, I'm gonna try to. Yep, I just pulled out my microtome. Okay, so see, that's actually one of the things I did not plan to do some, to cut the stem of Elodea with a microtome. But that's one of the nice things of live streams like this. If people actually write comments and we have requests, then I can of course respond to them. So let's change here again to the desk view. So this is basically what I got now. And I'm going to now try to cut. I'm now going to try to cut the stem. Oh, I forgot. See, here's an old one that I forgot. So, and I don't know. I'm going to just grab. I'm just gonna grab this here. I'm gonna just cut it, I guess. It's easy to cut, and I think I have to remove all of those leaves now. Oh, I broke it. It's so soft that it breaks too easily. So it's a little bit messy. It's gonna be fine, I guess. So I'll do all of the cleanup later. So the thing goes in here. Okay, that's my specimen holder for the Microtome. I 3D printed it. I have to try to squeeze it in here as well. Ah, didn't go. So let's try it again. The stem is very soft, which actually is a good thing because it should be easier to cut, but it's not long enough. Ah, okay. Another one, another try. Let's try to make it a little bit longer here. And you know what? You know what I'm gonna do now? Maybe I should cut this here. See, I think that's easier this way. Not rip it off, but actually cut it carefully. Yep. And one of the nice things about live streams like these are that sometimes I just improvise and try it the first time. For example, this is the first time I'm ever doing this. So let's have a look. So let's give this one here a try. And let's insert this. Here we go. Okay, haha, it's in. So, and it fell over. Okay. So what I'm gonna do now is, yeah, down here you can actually rotate this, and just move it in a little bit further. Okay. And we're now going to do the following. I'm going to just take my razor blade. Ooh, nice and easy cut. I forgot my glasses, unfortunately. So I'm going to turn it out a little bit. We're going to give it another try. Let's remove this. Ooh, look at this. This was actually quite nice. It was actually quite nice. I'm going to put it directly now on my slide. Here we go. This was actually a very easy one. Let's try it again. This was a little too thin. Also too thin. Yes, this was again good. I have to just remember where it is. Here we go. I think this is going to be enough. And I'm just going to add a drop of water now. And a cover glass goes on top. A cover glass goes on top. Yep. And let's have now a look at the cross section here. Okay. So this is, I have to, yeah, go back to the, here, that is the microscope view. I'm going to now remove, I'm going away here with that one, and I have to remove this here. And I'm going to now go and start again at the low magnification. I have to find the focus, of course. Here we go. Ooh, okay. I need to add a little bit. Yeah, I need to add more water. But this looks actually promising. Actually, I need to add a bit more water here. Here we go. Actually, it looks pretty nice. Looks okay. Let's go up with the magnification. Yep. Look. There seem to be those spaces in here. Looks actually quite nice. I have to admit, I'm positively surprised because normally making stem cross sections isn't the easiest thing to do because they end up very dark. Yeah, there are plenty of bubbles in here as well. Okay, but considering the fact that I basically have just did a very quick cross section, I'm actually pretty happy. Okay, yeah. So this is a question: is this live or is this alive? Well, this is a live stream. Yeah, in case yeah. So there seem to be vascular bundles in the classical sense are probably not necessary because it's a water plant. So it is able to absorb the water, of course, directly through the leaves. Yeah, there are plenty of yeah. But what we also see here, especially here, is that there are no chloroplasts present. Or at least not a lot. Maybe those tiny dots there, which kind of makes sense because those cells here are responsible for holding up the plant. Yeah. Quite cool. Still pretty nice. Yeah. The TV is talking back. Yes. Yeah. In case you're in case you're new to the live stream every Saturday except next week, unfortunately. I'm doing a live stream where I just show you a couple of specimens, and it's highly improvised live stream. This means that sometimes the things actually look nice. Sometimes there's some nice surprises, like the ones over here. I think this is a pretty nice surprise. I think it looks pretty nice. Elodea, cross section of a stem. Yeah, but okay, let's do a little bit of biology as well here. Okay, because. And I don't know, maybe some of my students are actually also watching here, but there's one thing that you'll notice, and so is that if you look on the outside here, yeah, of course you see the individual cells here, yeah, and on the outside here, what do you see? Nothing, and exactly that's the whole point, because many plants, land plants, what they have is they have a waxy cuticle on the outside, or a thick or an additional epidermis layer, which protects the plant from water loss. Of course, this is not necessary here, right? So you can actually see under the microscope as well that this plant is clearly a water plant simply because it's missing those protective layers on the outside. Yeah. Otherwise, yeah, because land plants obviously lose a lot of water. Yeah. Still stomata because of it's actually submerged in the water. It's actually floating in the water. Yeah, so what you have here, yeah. So and it actually grows also roots, but in the water. Yeah. So this is kind of So the concept of monocots and dicots I'm just seeing over here. These are basically belong to the flowering plants, land plants. Yeah. So look quite nice. Let's go down a little bit again. Yeah. So generally, I have to say I'm quite happy with the results here. Okay. And imagine if you if I were to spend a little bit more time in making the cuts a little bit more careful, maybe applying a little bit of vacuum to remove all of the bubbles that have accumulated in there, right? Yeah. Then, so what I'm going to do now is I am going to move on a little bit. I don't want to rush myself, but I do want to show you a few other things as well. Yeah. The name of the plant is Elodea densa or Egeria densa. It's a very, very common, how do you say, aquarium plant, freshwater aquarium plant. It's known to grow extremely quickly. Yeah. It's known to grow Elodea or Egeria. Both of them are related. Yeah. It's kind of interesting. I mean, if you go to an aquarium shop, they sell them to you, but actually once you've got them and if everything works out in your aquarium, they kind of grow so quickly that yeah. So let's do the following. Let's move on a little bit. How is that? But still, I really like this image. Makes me happy that yeah. That's a little bit this weird thing as well. It's, you see, microscopy is not only, I'm just cleaning up my table here. Microscopy is not only about science or anything. No, it's just observing nature and having a little bit of fun and then being happy that you're able to see something that looks nice. Yeah. So But sometimes it does help to understand a little bit the biological context. Yeah, then things simply make a little bit more sense, and sometimes, yeah, you wonder a little bit more about nature. Yeah. So let me clean up the trash here, just in case you're wondering what I've been doing here. I've been just cleaning up my mess a little bit. And let's move on now from a very nice looking specimen, like the one that you see over there, to a specimen that looks actually not nice. And that would be those the specimens that I extracted from the algae which I started last week. Okay, so that's the next thing, yeah, that I would like to show you. Again, more a proof of concept. Do not be too disappointed. Okay, it's not quite as nice looking. Yeah, so here it is. So what have I been doing? If you watched one of my previous videos, I've been 2 weeks ago or so, I visited a pond because I was on a trip with my kid, and I collected scratched off some material from a rock, right? This is the thing. What I have then done is I added something called hydrogen peroxide. Pretty aggressive hydrogen peroxide, 30% hydrogen peroxide, H2O2. You got to be really careful with that stuff. Really, don't get it into your eyes. It's highly oxidizing and very aggressive and very corrosive. And what it does is it oxidizes away all organic materials, and the inorganic cell walls of the diatoms basically are left over. So what you see over here actually is you see it's almost like, I don't know if I can show it to you over here. No, you don't see it well. No, maybe here. Yeah, you can actually see here on the bottom, it almost looks like a little bit like sand, right? Yeah, and these are indeed diatom shells and other things. And I just want to put that a little bit under the microscope right now. And the liquid that you have here on top, that is now water. Of course, I removed the hydrogen peroxide. Of course, it would be too dangerous. And I basically, washed it. Yeah. So what I'm going to do therefore is I'm going to take a little bit of this stuff here. And in case you're wondering why I've got some electronics here, I'm also going to tell you later. Yeah, it's microscopy related. It's microscopy related. Again, I'm going to change the tips here. I've 3D printed myself here a little tube holder as well. So, and I have done this already before with hydrogen peroxide, and I had some specimens that were really very nice looking, highly concentrated with diatoms, but in this case here, not quite as many as I hoped. It got a little stuck. Okay, it almost looks like very fine sand, and actually it is. Huh? Yeah. So let's do this. So again, this is a little bit more of a demonstration on how you can extract diatom shells yourself, but this one is probably not the nicest specimen. Okay, so you see that there's this white stuff here, right? It's kind of interesting because it used to be completely green and brown, and then you add hydrogen peroxide. Yeah, and I forgot to tell you, you don't only add the concentrated hydrogen peroxide, the 30% hydrogen, but you also put the tube into a warm water bath for a couple of hours. That's really important as well. It really speeds up the process. Yeah, so you add hydrogen peroxide to it. First of all, you remove the pond water, you add 30% H2O2, you put the whole tube with a cap on it into a water bath. And you don't get anything on your fingers or your eyes. Yeah, because this wouldn't be cool. Yeah. So let's have a look. So again, this is how it looks like. Again, not the nicest one, but yeah, it is a purification method, and we can actually see some diatom shells. Unfortunately, not as concentrated as I wanted to. However, the next cleaning step would be to add acid, like for example, hydrochloric acid would be best. I sometimes use concentrated acetic acid, vinegar. This would remove carbonates, and the cell walls of the diatoms are made of silica, so they are not dissolved away. Yeah. So what we have over here is a whole bunch of irregular stuff, but if you actually increase the magnification, you can actually see that they are also the diatom shells actually become visible. And again, I know I'm repeating myself. I've had prepared specimens where basically they were all over the place. That's a diatom shell over here. Sometimes I've also seen that some of the algae that were in there were not fully digested away by the H2O2. So you have to give it a little bit more time. Here, that's a fragment of a diatom shell. This one down over here are a few. Here, this one. So again, you can see them much better. Over here there is a nice one. There are two of them here. So what I'm going to do is here, and here. Let's go up with the magnification. So what I'm going to do, of course, is I'm going to continue processing it, and I'm going to add now a little bit of acid to actually hope that many of those things are going to be removed, because they might be carbonates and other things. And the carbonates, they will calcium carbonate and so on. They're going to be removed by the acid. Some of that's, for example, a long diatom shell over here. Okay. You're wondering about the electronics here. No, it's not galvanotaxis. It's not galvanotaxis. So I just some kind of a crystal here. I don't know what it is, but again, this is more a, how do you say, a proof of concept that I just wanted to share with you. But again, I know that they're probably not nicer specimens. So I have to honestly admit I was a little bit disappointed with this one, but it still shows that it kind of works. Okay, so there is we still have 20 minutes or so. So what I would like to do now is I would like to how do you say, resolve a little mystery here that I have. That some of you are kind of wondering. Is what in the world? What's going on with this here? And I'll just share this with you, but I'm going to tell you that I am working on a separate video that I'm going to be uploading also to this channel in the next, I don't know, maybe a week or two. I still need to do a little bit more filming. It's going to be a slightly more technical video, not an observation video. This is I'm going to show you now a power supply of a stereo microscope. Now you wonder, why in the world do I have it here? I'm going to show you just a second the stereo microscope. Not the complete one, but just the base. Here it is. Okay. So and there's supposed to be a lamp in here. That is the top illumination. It's kind of the green screen is kind of acting up a little bit here, right? Or you know what? I'm just going to put it in here. And there's the halogen lamp is supposed to be here. And now I'm going to show you the problem that this microscope had. As a matter of fact, there was, and if you turn it around, then I can also show you in this model here, where there's a transformer here, and this is the power supply. So basically, of a second microscope, exactly this one over here, right? It's in here, right? So why did I take it out? And I just want to share this with you in case you have a similar problem. The problem is that we have now we have 15 of those microscopes in school where I teach, and they're in very good condition. No problems with those. However, let me put it down. I tested them yesterday, and all of the 16 had a problem, and that the lamp, both lamps were flickering. Brighter, dimmer, brighter, dimmer. And this is so annoying. And I did a little bit of research, and I found out that the reason why the lamps of the microscopes are flickering is because those capacitors over here are too old, and they have to be exchanged. So what I've done is I've done a little soldering project, and I bought myself, and it worked. That's the good news. I bought myself a couple of capacitors here, where I cannot open the thing anymore because it's so tightly closed. Oh, here we go. So I bought myself some capacitors, 50 of those. Okay, here they are. To exchange those. So I soldered them out and I replaced them with new ones. And it works. Okay, at least those microscopes that I fixed this way, there is no more flickering. And some of the flickering was pretty bad. And yeah, so this was indeed the problem. And why am I sharing this with you? Is it kind of shows a couple of issues, how important it is that microscopes are made in such a way that they can be serviced. That's kind of my message. So microscope manufacturers out there, if you anyone of you is listening, please take a good example on this microscope over here, that it can be serviced. Okay, at least some parts can be exchanged. My request, however, is to make it even more serviceable, because some of the parts, there's one microscope where one of those voltage controllers is broken. I can't get the parts. So that's a frustrating thing, and we have to be aware that especially in if the microscopes are all kind of closed off, and if there are no parts available or too difficult to disassemble, then this can be an issue. And I have to tell you, this is going to be a little bit where I'm going to be busy a little bit. I'm going to be start fixing microscopes, and I'm going to be exchanging those capacitors. So remember, flickering lights in a microscope, most likely a power supply problem. Yeah. The thing is, why they are flickering? They're flickering because it is normal for capacitors to age. They will actually start breaking down even if the microscopes are not in use. Yeah. So this is a little bit a general problem, and this is actually kind of weird. Not weird is not the right thing. You see, some people buy microscopes because we want them to last forever, right? I mean, especially, but the weakest part, what I sometimes find of a microscope, is not the optics or anything or the mechanics, but it's the electronics, right? So because with heat and turning it on, turning it off, and all these things, of course, the electronics may suffer. I'm kind of happy that at least the fix is fairly easy. Yeah. So just wanted to share that with you a little bit. I'm just putting this away so that just wanted to share this with you a little bit. Yeah. And I've got something else here that I want to show you, and I have not looked at it myself yet either. That's this here. Okay, let me open this here as well. This is vinegar, and actually pretty old vinegar. And there is actually some stuff floating in there. And these are microorganisms, probably bacteria or maybe a mix of different, and they basically are able to live in this acid. And this one here is pretty bad. I used to grow my vinegar eels, nematode worms in here. They all died. But if you actually look at this, there is some solid stuff in here on the bottom. I don't know if you're able to see that. That is something that I want to put under the microscope. Maybe a collection of dead worms, bacteria, and other cells. I don't know. But there seems to be something growing in here. And that is something that I would like to investigate. Because as a matter of fact, vinegar, there are certain bacteria that grow in vinegar which are actually converting alcohol also to vinegar. So these bacteria make vinegar, but these kind of formed afterwards. So I'd like to know what it is. And really, I did not look at it yet before. So it could be indeed that there is nothing exciting either. So we're going to find out. And I'm just reading as well. Changing out capacitors in older equipment is very common. Yep, those electrolytic capacitors, apparently some of them are also able to explode. This is disgusting. I love it. Look at this slime. Slime means polysaccharide. This is a lot. Okay, look at this disgusting thing here. Yeah, yuck. I don't know. We're gonna see. We're gonna see what's in here. This is all, I don't know. There's almost the whole sample here that I've got. So, occasionally I'm reading here. Yeah. Yeah, the polarity of the capacitors is important. I know. Otherwise, if you put them in the wrong way, so the capacitors, they're all 1,000 microfarads, and you have to also take care that they're able to withstand the voltage. So the capacitors that were in there were 16 volts and 25 volts, and I'll replace them all with 25 volts. Thank you. Here are the questions. Okay, which I'll quickly go through. And let's put this on here. Look at this. Squeeze it flat. Yeah. Every now and then, if there's too much liquid, yeah, this one seems to be doing okay. But especially when you want to look at bacteria, assuming that these are bacteria, you really must make sure that the how do you say, the layer of liquid is very thin, because you want to restrict the movement of the bacteria if there are any in here at all. So let's kind of soak up some of the vinegar here. And it goes under the microscope, and as always, the hour of truth, because I don't know what to expect. The hour of truth. I need to change again to the microscope view here. Okay, too bright. This is an interesting one. So sometimes if there are any bacteria or whatever present, or fungi present, then sometimes they're sticking together so much that you're not even able to see the individual cells here. Let's have a look. Oh yeah, those tiny dots that you see, these might be actually bacteria, as expected. And here again, I'm kind of always surprised. I mean, I know nature is very diverse, but some organisms are able to withstand quite some acid. So these are definitely bacteria, those tiny things here. Yeah. Yes, the question about cleaning the oil objective. I'll clean them. Just a second. I'll do that later. We do the following now. I'm going to switch over to 40 times, and I'm going to go and into phase contrast. And then we're going to see it better a little bit. This is kind of not so nice. Why is this? Okay. You see, it is. I tell you, it's not centered. What? Yeah, here I have to turn a couple of screws here to get everything. Looks almost a little bit like oblique illumination because it's not properly centered. Yeah. Maybe the other one is. Yeah, it's a whole slime matrix what we have here. But you can see that there are a couple of dots here that are the tiny one over here. So there is actually some movement going on here as well. Here it's chasing it. Yeah. Now this is now some kind of a liquid flowing. Yeah, so those when you see actually some kind of slime sticking together, then very often the cells are secreting some maybe polysaccharide or something, yeah, which causes them to stick together, and that's why it looks the way it does. But again, yeah, at least now I can dump out this vinegar with good conscience, also having looked at it. And those tiny things that you see, of course, all bacteria. Okay, not so super exciting, but at least, yeah, we now know that there's stuff growing on here. The other one over here is actually probably more, I don't know. Let's have a look at the other one over here. And there is a lot of this stuff here on the bottom. Go all the way in. Look at this. Oh. It is disgusting, of course. So I'm using a large cover glass now to spread it very thin. Yeah. Yeah, this is also very yeah, not so nice looking. Let's have a look again. I'm going to just look at it again using my low power. Ah, wrong. Oh, this looks more interesting. Okay. Yeah, but those patterns that you see here, that's because also because it was kind of the organisms were kind of growing in sheets, and therefore they kind of folded on itself. That's why it looks the way it does. And yeah. Yeah, so basically, or maybe, yeah, cells that are sticking together are always difficult to see because they kind of form this uniform, homogeneous layer. Maybe those tiny dots could be individual cells. Ah, okay. Pretty boring. I think I don't feel like spending more time just observing this there. I prefer the Elodea. Yeah, so basically, I don't know if you've some of you have, are making kombucha, for example, or SCOBY, you know, the fungus. That's kind of the same. You have this fungus and sometimes also bacteria. It's a yeah, gigantic colony that then will ferment whatever is in there, yeah, in the drink. Yeah, and you have those also forming now in the vinegar. Yeah. Let me see if maybe over here. Yeah, also all those tiny dots here. The bacterial concentration is so high that you're not even able to see the individual cells here. Let me see, maybe we can move a little bit further away here. Let's turn up the light. Yep, it's better here. See if there are, see when there are fewer, then we can actually see them better. Here we go. That's what I was hoping for. Look, here, there are fewer over here. And then we're able to see these are definitely rods. It's tiny, dark. That's better. Yeah. So here we go. Let's do the following. And yep, that's okay. Yeah, those tiny little things that you see here, these are the bacteria. So the reason why we could not see them before, because there were simply too many of them. Yeah. And yeah, they kind of must have fallen into this jar of vinegar, and then they started to grow there. And I think what's important, however, also is that of course different environments prefer different bacteria. So this, or select for different bacteria. So this means that of course only those bacteria can multiply that are sufficiently acid tolerant. Yeah. Bacteria, my favorite flavor. How do bacteria taste like? Well, it depends on the bacteria. You've got them growing on cheese, then they taste like cheese. You've got them in yogurt. So biotechnologically, they're pretty important. Yeah. So, okay. Oh, I'm already talking for an hour, and I said I'm going to quit after an hour. But no, I will now do the following. I'm going to quickly go through a couple of the questions here. The first one. Did Mini Microbehunter win his chess matches in the previous outing? The reason why I was not doing a live stream last week is because I went with my son to a chess match, and yes, he won both of them. He was competing in the Austrian national competitions, not for kids, but actually also for adults. And yeah, he was. But these were basically only 2 rounds. So it would be cool to see the Elodea stem. I have done that. under the right conditions, could white blood cells live outside of the body? Short answer, no. More complex answer is yes, I have already put my blood under the microscope, and you can actually observe the white blood cells moving around for some time. But they will not be generally able to live outside the body. Look, if the blood sample is sufficiently large, and if, for example, like in the blood, how do you say in hospitals when you take blood samples and they store the blood, of course they have to survive, right? No, I correct myself. I completely correct myself. Of course, they can survive if you make the right conditions. And for example, this is what doctors are doing. You're growing white blood cells that you extracted from your bone marrow for stem cell treatment. Yeah, so that's what they're doing. Yeah, if the conditions are right. So I know I have to completely correct myself. Because if you make the right conditions, then the cells don't know that they're outside of the body, right? So the temperature has to match, the nutrients have to match, and of course you're growing them in the lab. Yeah. So are chloroplasts alive? Yeah, that's a question of definition that I was talking about. Off topic, future video idea looking at scratched surfaces, particularly metal, comparing natural light-worn surfaces like an old, well-used teaspoon versus deliberate and deeper scratches to see differences in reflections with different angles of light. This is something which I can do with my stereo microscope. Okay, thank you for the recommendation. The spontaneous live stream is best, thank you. Still stomata because the part of the plant would be above water. Elodea generally not, because they are actually drifting in water. There are no vascular bundles. Yes, we talked about that. Is there a micro technique to take out the water and replace it with resin, like von Hagen? Yeah, von Hagen. But in Microscope we could make permanent mounds of water plants. Gunther von Hagen, he was the guy who is the German who basically has been plastinizing human bodies. It was, I remember back in 30 years ago when they started to have the first exhibitions, it was considered quite controversial. But the idea is the following: is are you able to do something similar with water plants? And the short answer is yes. Of course you can do that, and this is actually also done. Also in histology, what you do is you have to carefully dehydrate the plant using an alcohol series, and then you have to embed it in a resin, not in a resin, but in paraffin, and then you can microtome it. And as a matter of fact, yes, this is actually a way how you can make permanently mounted slides. So this is actually done, also with a human body. Right, you make when you take tissue samples, you have to dehydrate it first, and then you have to take out the water. You have to put them in paraffin, you microtome it, and so on. However, this is I would say a pretty elaborate process. I've not done this so much yet. I've tried it a little bit, but it's a highly elaborate process. But it is possible. Yeah. Off topic, what was the name of the thick white stem you usually do cuts off? Some kind of a lily. This was a tulip. Yeah, I'm not noting down the names of what okay. I'm noting down. So basically, the stem that I cut some time ago is what you always want to use: stems that are not woody. Right, not wood, don't contain the lignin. I've basically observed also sunflower stems look quite nice looking, or also tulips, and they are fairly large stems. And what I've done is I cut them, and you can simply place them in concentrated alcohol, and you can store them in that concentrated alcohol practically indefinitely. So I've got some samples somewhere in my cupboard, which I already kept there for 25 years. I'm not kidding. It's a little bit like a souvenir. Electronics in microscopy would be super cool to see. It would be great to learn a bit of engineering personality with. Okay, yes. I'm gonna make a video on how I fix the microscopes, and maybe I'm gonna make a few more videos on this. However, I'm not going to do a detailed deep dive and tutorial on how to fix microscopes, electronics, because every microscope is different, and I still do want to kind of stick to the observation part. But I do know that fixing microscopes is actually also pretty important. Yeah. So What I'm going to do very quickly right now is I'm going to quickly run through the chat here in case there's some comments that I forgot. Okay. No. Whether chloroplasts are alive, that's an interesting one. Yeah. Future video ideas, scratches. You can use a bright flashlight to get decent epi or top illumination. That is correct. If you use the four times objective, okay, like this one over here, yeah. You check up here, then there's still 2-3 centimeters distance, and this means that you can use a flashlight, a strong one, shine it on top, and then you're also able to see the surface texture. Yeah. So, yeah. Okay. Yep, I'm reading. Yeah, classification. I did bacterial when at my university studies. I was doing bacterial identification. That's a complex one, I tell you, because people put living things into categories, into boxes, and later on you discover that the categories were actually wrong. So I forgot. Here we are again. Okay. So what I'm going to do, I think that I pretty much went through everything, and we're closer to higher resolution with a compound microscope. Do you use lens fluid for cleaning your oil objective or just lens paper? Only lens paper. Okay. Thank you for reminding me, because maybe that's the last thing that I'm gonna do is I want to kind of simply do now the following. Look, I'm gonna try something now. Just a second. I'm going to try something now. I'm gonna try something now. I'm going to remove this here. Just a second. How does this work? Yes. I'm going to now do the following. Are you able to see this? Yes. Okay. Yeah. I made this a little bit bigger now. And what I do now is the following. Here, this is now the oil immersion objective. I remove the slides. You see, actually, this camera is kind of slow for the stage. And then what I have is the following. I lost it. Where is it? Where did I put my lens paper? I just had it a minute ago. Here it is. Here it is. Okay. So this is lens paper. And what I usually do is the following. I take a lens paper. Here it is. Yeah. And I rip off a small piece like this. Yeah. And then I just fold it. And I simply carefully go here to remove the oil. Okay. And that's it. See over here that it's oil. Yeah. The oil is here, and then sometimes I do it again, maybe just to make sure that it's dry. And there are different opinions about this. Some people are using, I've read in forums, they're basically using the oil immersion objective on a permanent basis every day because they're doing this as a job, and they say they rarely clean it. Okay, because it's always... however, if you're using the natural immersion oil, which is the yellow one, then actually it can become hard over time. And you should therefore remove it, right? Because otherwise it might actually stick to the lens. And another thing is because as long as there is oil on here, it might actually collect dust as well, because the oil is sticky, and this might also not be the best thing. Okay, so this is simply something. So, and then I only use this once, and I throw it away, because after a couple of minutes, the oil is going to spread all over the tissue paper. Right. I talked to the representative of the company. And he said, told me the following. He told me the following, that people sometimes are too afraid to clean microscope objectives, and that many of them are designed to actually withstand more. Yeah, you can actually go in with your fingernail to actually scratch out the dirt a little bit if it becomes dirty. He told me. Yeah, so it's not they're able to withstand probably more than what we fear, right? So the thing is lens fluid. no, don't use any solvent is necessary. You can do the following. Some people are using alcohol, concentrated alcohol, but you have to be always careful when you're using liquids. I mean, then they might actually go into the cracks. What is more dangerous, I would say, is that if there is actually oil going into on the other lenses, right? Yeah. Yeah. So that's yeah. So this is something that you also have to clean away. Yeah. So, ladies and gentlemen, I hope that you liked it. Yeah, I think I'm going to call it a day today. I hope that I was able to answer more or less all of the questions here. Olympus lens paper, it's actually not from Olympus, but yeah, any lens paper will do, right? But in any case, you should not be using any commercial liquids like liquids for cleaning glasses and so on. You should not be using. Okay, so yeah, I'm going to call it a day today. I hope that you liked the live stream. Yes, again, next week I have to basically skip it again. Next Saturday, but I hope to be able to see you again. And of course, please check this channel. I'm going to be uploading again an Elodea video, probably tomorrow. I want to wish you all have a nice day and happy microbe hunting as always, and see you again. Bye bye.